Transformation is the genetic change in a bacteria or a yeast as a result of a penetration into its core of a DNA molecule, while transfection is the introduction of foreign genetic material into eukaryotic cells by plasmids, viral vectors, or other transfer tools. Transfection can be induced by electroporation and sonoporation. To choose the best method for transforming bacteria should consider the following aspects. Very small amounts of DNA are required into transformation, about 5 to 100 nanograms, because excessive amounts of DNA, which means over 100 nanograms, inhibits the transformation process. In other words, the transformation efficiency is inversely proportional to the size of the plasmid. Bacteria are required to be in a specific physiological state called competition, in which bacterial walls are permeable to macromolecules made of DNA or plasmid specifically. The amount of salts in the DNA, the structure of the cell wall of the bacteria, and the resistance of the organism to certain temperatures are also important to consider. Besides heat stock, electroporation, and sonoporation, there are other methods to transform bacteria like biolistic, which uses heavy metals like gold or tungsten, which are coated with DNA and shot into the plants with the help of gene guns, commonly used to allow the transformation of plants' plasmids. Viral transformation or use bacterium-mediated transformation.
The chemocompetent cells had 11 colony-forming units and a transformation efficiency of 2.2 times 10 to the third colony-forming units per micrograms of PRSET. The electrocompetent cells were 14 colony-forming units and an efficiency of 2.8 times 10 to the third colony-forming units per microgram of PRSET. The basis for the transformation with chemocompetent cells is the creation of momentary pores in the cell wall by the action of cations like calcium or rubidium by the interaction with the negative phosphate groups in the plasmids and the cell wall. Applying also a thermal difference, this procedure takes place. On the other hand, electroporation consists in the momentary pores achieved by the effect of an electric field. The electroporator requires a special cell with metal plates to allow a better flow of the electrons. In this practice, 1500 volts, 25 microfarads and 200 ohms were applied for 5.2 microseconds. In both techniques, a highly pure sample of DNA with its correct concentration is imperative for an efficient transformation. For example, salt contamination from the DNA sample in electroporation can cause cell lysis. The efficiencies obtained are extremely low. It is estimated that a good efficiency to be greater than 1 times 10 to the 6th colony-forming units per microgram. For instance, New England Biolabs 5-alpha has a reported efficiency of 3 times 10 to the 6th colony-forming units per microgram of PUC19. The greatest efficiency was 2.8 times 10 to the third colony-forming units per microgram of PRSET. The cells do not have a great ability to be used for transformation with supercooled plasmid, even less likely for ligation products. The electrocompetent cells could have been put through to a very aggressive electric field and died. The chemocompetent cells could have been submitted to inappropriate concentrations of calcium chloride. In both cases, the amount of plasmid could have been a limiting factor.
There are two principal differences between the chemocompetent and electrocompetent cell transformation methods. First is the presence of salts in the chemical method. In this case, we use calcium chloride in order to neutralize the phosphate groups in the lipids of the cell membrane, which repel the negative charge of the phosphates in the DNA. The second difference is that in electrocompetent method, we need to add 10% cold glycerol in order to avoid any contamination with salts. We obtain a chemocompetent efficiency of 2.2 times cubed and an electrocompetent efficiency of 2.8 times cubed. These efficiencies are lower than expected. Normally, the electrocompetent cells have an efficiency of 1 times at the ninth power and the chemiocompetent is about 1 times 10 at the sixth power. These cells still can be useful for plasmid ligation, but we don't expect high results.